Bismillahirrahmanirrahim. Today we are going to deal with let's get the laser pointer to deal with the preparation of tissue for electron microscope. What are the learning outcomes today? Number one, to describe the steps of preparation of sections for electron microscope. دي أول حاجة steps وهي بالظبط زي المايكرو تكنيك اوف بارافين بارافين تكنيك. To recall the steps of preparation for paraffin وهنحاول برضه نفتكرها كالعادة. List the fixatives used in this technique. Memorize the stains used in electron microscope and write the types of electron microscope and to compare between light and electron microscope. Calculate the magnifications of light and electron microscope. Let's start. Yes. Types of microscopes. We have many types of microscope, like light microscope that you utilize the light for visualizations of the sample. Electron microscope, here we depend upon the electron beam for magnification. Fluorescent microscope, we use the fluorescent light for visualization of the sample. Phase contrast microscope and confocal microscope, which utilize the laser for visualization of the sample. Measurements in histological study, 1 centimeter equal, yes, how many number of the, yes, 10 to the power of minus, yes, 2, yes, 10 to the power of minus 2 of the meter. 1 millimeter, 10 to the power of minus 3 of the meter. 1 micrometer, 10 to the power of minus 6 of the meter, هو 1 على مليون. 1 نانومتر 1 على مليار يعني 10 to the power of minus 9. These are the units in the histology. This is the light microscope. Here I have the eyepiece, objective lenses, nose piece. This is the slide. This is the stage holding the slide holder, and the key of the this is the key for the movement of the slide. This is the diaphragm and the condenser. This is the focus adjustment, coarse adjustment, and a fine adjustment. The laser, the light, the source of light, and this is the base, and this is the arm of the microscope. Yes, these are the components of the light microscope, essential components, yes, essential parts of light microscope. Magnification of the power of light microscope. We obtain it through the multiplying the power of the objective lens with the power of the eyepiece. And if the objective lens is 40 has 40 magnification power and the eyepiece 10 magnification power, so the magnification power of the light microscope will be, yes, 400 magnification power. Preparation of tissue for electron microscope. You will know that it follows the same rules of the paraffin technique, starting by sampling of tissues 1 millimeter. It is smaller than paraffin and smaller than frozen. In paraffin, we are less than 1 centimeter, mostly 5 millimeter. In frozen, we have 2 by 2 millimeter. And here in electron microscopic examination, we have a sample of 1 millimeter, only 1 millimeter, no more than 1 millimeter. Add the glutaraldehyde and osmic acid, penetrate only half millimeter. So to ensure the reach and penetration of the glutaraldehyde and osmic acid to the whole sample, we have to sample only 1 millimeter. Fixation, we have double fixation here, glutaraldehyde 4% followed by osmic acid 1%. And the action of osmic acid is not only fixative, but also it will stain the fat by stain, which gives the fat black coloration, especially the cell membrane and organelle membrane. Dehydration by ascending rates of alcohol, as usually we accustomed in the paraffin technique. Dehydration starting by 50% alcohol, 70%, 90%, and absolute alcohol Then clearing by propylene oxide in step number 3. Remember, we use xylene in the paraffin technique, embedding in the plastic media, which is the epoxy resin and the oven also, but it takes time more than 4 hours. This step is very, very critical. Sectioning by using ultra microtome to cut thin sections at a thickness of 60 to 80 nanometer or 50 to 70 nanometer. Staining here using heavy metal salts like uranyl acetate and lead citrate, This is the ultra microtome cutting at thickness of 50 to 70 nanometer. Activity compare between the 3 techniques, paraffin, electron, and frozen technique, as regard sampling, dehydration, clearing, impregnation, embedding, cutting, mounting, staining, and examination. Electron microscope, types of electron microscope. Here we will know the types of electron microscope. We have transmission electron microscope to visualize the internal structure of the cell, and scanning electron microscope to visualize the surface of the cell. This needs surface, so it depends on scanning and scanning coil to give 3-dimensional image on the screen, as we will see. Transmission electron microscope, the source of illumination here is the beam of electrons emitted from the heated tungsten filament, and the beam of electrons are attracted to the anode at a set of electromagnetic lenses. The beam of lenses passes through the series of electromagnetic lens which serve the same function as the glass lens of light microscope. يعني بنبدل هنا بنستعمل في اللايت مايكروسكوب بستعمل اللايت وبستعمل الجلاس لينس, while in the electron microscope I will use the electrons and I will use the electromagnetic lens. Also we use condenser objective and projector electromagnetic lenses. The final image is received not by the eye as in light microscope. We receive it and view it by fluorescent screen or on a photographic plate. This is the electron microscope, beam of electron, electromagnetic field, condenser, electromagnetic objective lens, and this is the projection lens, and this is the screen image. Where I will put the specimen will put here like this. Don't forget that we cut or section the specimen at a thickness of 50 to 70 nanometer. This is very, very important to compare between the paraffin technique and the electron microscopic technique. This is the body and the whole instrument of the electron microscope. You will find that here I will put, this is the electron beam source, the anode and the cathode, and this is the multiple electromagnetic field act as a lens. And here I will receive the image at the fluorescent screen, and to edit this image and photograph it and save it through the computer screen and computer PC. Magnification power of the and resolution power of the EM or electron microscope from 1,000 to 100,000 to compare with the 1,000 magnification of the light microscope. So it had enormous magnification power reaching to 100,000 magnification. Resolution power is 0.2 nanometer, 0.2 nanometer. What is the resolution power of naked eye? Naked eye, 2. Yes, 0.2 millimeter. Resolution power of the light microscope, 0.2 micrometer. Resolution power of the electron microscope, 0.2 nanometer. The terms of electron microscope, we will find that we use electron dense, which appear black, electron lucent, which appear gray or white gray. Scanning electron microscope similar to the electron microscope, except it has scanning coils that scan surfaces, and project the electrons to electron detection system, and the system project emits images to special computer program for interpretation and recording. This is the scanning electron microscope. You will find here anode and the electron beam, electron source, and this is the condenser electromagnetic field. Notice here the presence of scanning coils, which will scan the sample, and another secondary electron detector, which analyze this sample or the electrons coming from the sample to determine the sources of the sample. This is the body of the scanning electron microscope. Notice here the surfaces of the cells or surfaces of the images appear on the BC of the scanning electron microscope. To compare between light and electron microscope, as regards source, here's a source light, while here electron, lenses, glass lenses, electromagnetic lens, image eye in on fluorescent screen, resolution 0.2 micrometer, 0.2 nanometer, magnification power up to 2000. This is in the confocal microscope, while here from 1000 to 10 or to 100,000, mounting on the glass, while here in copper grade. This is the two types of electron microscope. I have the transmission electron microscope here, while here the same image by scanning you will find the surfaces. This is the transmission electron microscope, and this is the scanning electron microscope. Assignment questions compare between paraffin and electron technique as regards sampling, fixation, dehydration, clearing, and bearing, cutting, staining. List 4 types of electron of microscopes. You remember light, electron, fluorescent, confocal, phase contrast. Calculate the magnification power of light microscope when we use eyepiece 10, magnification power 10, and objective 100, so the magnification power will be 1000. What is the correct sequence of electron microscopic technique here? Yes, starting by sampling, fixation, dehydration, clearing, same as paraffin. Chemical using clearing tissue before impregnation in paraffin, yes, xylol. Chemical used in clearing tissue before impregnation in epoxy resin, yes, propylene oxide, yes, this is number B. To compare between epoxy resin, propylene oxide, while in paraffin we will use the xylol, yes. Temperature of the oven during paraffin technique, 55 degrees Celsius. Which of the following tools used in sectioning of electron microscopic technique? Yes, no, cryostat wrong, microtome wrong, ultra microtome is the correct answer, number C. Which of the following stains used for electron microscopic technique? Yes, uranyl acetate. Yes, uranyl acetate. Number E is the correct. Sectioning of ultra microtome is usually at a thickness of 50 to 70 nanometer. Don't forget the unit, please. Which of the following embedding media is best used for sectioning of ultra microtome? Yes, embedding media. Yes, epoxy resin. Yes, epoxy resin. The best range of temperature of cryostat is minus number A, minus 13 to minus 20. Define the type of microtome in the following photos. Yes, what is this? Scanning electron microscope. What is this? Transmission electron microscope. What is this? Scanning electron microscope. What is this? Transmission electron microscope. Scanning electron microscope. Transmission. Scanning. Scanning again. Thank you for all of you. See you again. Many thanks for all of you. Assalamu alaikum.